everybody. First of all, thank you for the invitation. Thanks to GRIN Europe for preparing the meeting, this amazing program, and special thanks to GRIN Italia for hosting this meeting and changing the scenario. So it's very beautiful scenario and very happy to be in a growing audience. Okay, so in the next 20 minutes, I'm going to try to catch up with the new updates on the progresses performed in the Barcelona team that is composed mostly by wet lab people, as in my lab, computational people, as in Mireya's laboratory, and the clinicians. So I think that one of the messages of my talk is that it's very important to join efforts and to have this multidisciplinarity, especially in the field of GRIN disorders. Okay, so this is a reused picture, and this is how we see GRIN disorders. So all GRIN variants there are hundreds, probably thousands of these variants. When looking at the universe, we are fascinated by how they bright, but at the same time, it's a big challenge to understand which one is producing an effect, or how we can treat all of these, how we can explore, how we can understand. So, in order to address this big challenge, all the people that is in this room, we are doing many overlapping techniques, and today I prepared an outline because I want to be structured, okay? So the first one is about the functional stratification. I very briefly introduce you a new method that we are proposing to study those GRIN variants that are in the gray zone, or they have no technical strategies to stratify. In the second part, I will introduce you our drug discovery approach, very focused on a small library, and I will finish by presenting a paper that we recently published on the use of a new molecule for treating GRIN to be loss of function variants. Okay, so we started with this, trying to understand this galaxy of GRIN variants, building up a GRIN variants database. This was initiated and is currently performed by Mireia Oliveira. She generated the GRIN database that is complementary to the others that have been already described, the one from Leipzig, Emory, and the GRIN portal. And we recently updated these, so there are the number of variants is tremendously increasing. You can take a look, especially that not all the variants of GRIN or GRIA are pathogenic. So with HEC cells, we expected that no changes would be occurring. But when we look at the synaptic activity, in that case, the EPSCs, the excitatory postsynaptic currents, we observed that the peak amplitude was reduced as well as the area, indicating that potentially this is a loss of function variant. Of course, we want to use these models also to try to rescue using pharmacological strategies. Indeed, we want to apply either a positive allosteric modulator or a potentiator or an inhibitor to see whether we can rescue. And it will be the second validation of this functional stratification. We have also this one, the serine 617 to cysteine, that was classified in our lab as a complex one because it provoked a diminished current amplitude, but it was not blocked by magnesium. So we were in this gray zone, and when we expressed in neurons, we observed that, again, this one seems to be a loss of function. So this is like a new approach that we are developing. It's still under progress, but I think it can provide a good tool to stratify these orphan variants. Okay, so what about the new potential compounds? So basically our rationale is very simple. We have a different GRIN variants and GRIN genes that assemble together the gene products. We have this spectrum of NMDA receptors activity that can be downregulated or increased, and we want to provide compounds that will restore this, what we call homeostasis, this balance, this normal range of NMDA receptors activity. So we wanted to identify novel compounds or use compounds that were described in the literature and to adapt to our research and try to rescue the synaptic deficits, the current alterations, and the behavioral tasks in our mouse models. So this was already published. We started with L-serine. I think that all the audience knows that. So L-serine is a precursor of D-serine, which is the coagonist of the NMDA receptors. We validated using cell lines, cellular models, more simple, or also in neurons. And then we performed a clinical trial, the first clinical trial that was conducted by Ángel García Cazorla in Barcelona. And importantly, these results were also confirmed by other groups, such as the one in Leipzig or also in the Netherlands. So that was, we were very happy about this, and this is the first line treatment for GRIN loss of function individuals. But still, something is missing. It's not a complete rescue, and we need to develop additional tools. So the rationale is still the same. We want to identify compounds that are able to restore this normal function of GRIN variants. And together with Mireia, she was developing an in silico docking strategy. So for a lay description of this is basically to place the receptor in front of all the drugs that are described and to see which of those compounds, metabolites, drugs can fit in the structure and potentially could modulate. So using this approach, we have a very short library of compounds. So we have 12 compounds that we are evaluating by different means. So first we are starting using cellular models and then more and more sophisticated models such as in vivo models, moving to ex vivo or also in vivo. So I'm going to give you some insights on the new compounds that we are characterizing. We have two of them, AM2, AM4, that are increasing the activity of the NMDA receptors. This is done in wild-type receptors, okay? We are not looking at the mutations at that time, so the rationale is to work for those mutations that trigger a loss of function. We want to potentiate, of course, these subtype of receptors, but also the wild types, which are present in the synapse as well. So we have been incubating these compounds with the cells expressing these receptors, and we have identified AM2 as a potentiator of GLUN2A containing receptors and also GLUN2B. And for AM4, we have seen that this effect is only observed in GLUN2B, suggesting that this compound could have a best fit, the binding or the efficiency of its effect will be more specific for GLUN2B. And this is important for pharmacology. We have another compound from this short library. So this is, this point is not published, it's like very new, but I wanted to share today. So this AM3 compound is very interesting. We have seen that although the current amplitude is not affected by the administration of AM3, we observe that the kinetics, the desensitization rate is accelerated, meaning that the channel is open less time, and then there is less current conductance. So we have, this was in hex cells, so in cell lines. We moved to more sophisticated models in the primary cultures. So we are currently developing, you can see these neurons that are like beating. We load with calcium indicators, and we can observe the general activation of these neurons in response to AM3. And we have confirmed that in primary neurons, it is reducing also the current amplitude, and we are still waiting for the results of calcium imaging. This compound, we have been doing like all the characterization, but this is in progress. I have been observing that ex vivo, so when we extract one part of the brain of the mice, we can treat this brain slice, this hippocampal brain slice, with AM3, and we reduced the excitatory currents. And also when infusing, and this is an in vivo experiment, when infusing the compound and registering the activity of neurons on the hippocampus in the alive animal, we also observed that it was having an effect. This is not exactly for this compound, sorry. So this is not for AM3, but we have been setting up this protocol to evaluate whether AM3 would reduce this activation. And to end up, I would like to show you the work with spermidine. If I still have 5 minutes, I think that I'm running. No? Yeah. Yes. And to show you the results on spermidine, so this is something that a work that we have completed, at least the proof of concept study, and I'm very cautious to say that I'm going to show you a proof of concept study in 2 patients, okay? But it's quite promising. So this was, in that case, we used this literature approach that was based on what was known about the regulation of the NMDA receptors. And especially the works of Pierre Paoletti and Laetitia Monnier in Paris, they were working with spermidine and spermine, okay? So spermine binds to the NMDA receptors and provides a positive allosteric modulatory effect, so potentially it could work for loss of function variants. So this is the history on how spermidine was identified in the 17th century and the proposed model. I'm going to skip this, but it's pretty fun, so if you have curiosity on the experiments from Leeuwenhoek, you will understand the etymology of the word spermine. Okay, so spermidine has been used for different clinical issues. It exerts a cardioprotection effect. It has been proposed as being a cognitive enhancer. And at least for us, we have evidences that spermidine can be converted into spermine, so increase the levels of spermine in brain, and is a safe compound. So with this data in hand, we moved to the lab. We evaluated what was the effect of spermine. So this trace was in absence of spermine, and when we treated with spermine, we observed a potentiation that was cool for us. Also for the mutant, this loss of function mutation was also increased. And when comparing the effect in vitro, we observed that spermine effect was much higher as D-serine. So this data suggested that spermine could be a more efficient potentiator of the NMDA receptors. We confirmed this in different mutations we expressed in heterologous systems, and we observed that spermine was in all these mutations increasing the activity of the mutant NMDA receptors. We moved to primary neurons. We observed that this potentiation resulted in an amelioration of the spine's density and a normal, an increase of the total spine's density, especially the stubby neurons. And then we moved to the mice. So we performed this battery of behavioral and molecular and electrophysiological characterization that we described, others described this morning. Today I'm going to focus on the GRIN2B heterozygous mouse model, which is a loss of function model, but also we have been characterizing a GRIN2A loss of function and a knock-in that we recently generated. So this is the battery of tests that we performed. Remember, the rationale was to introduce spermidine in the diet of the mice, so in that case in the water, but in children in form of pills, of a supplement. And this spermidine will be metabolized into spermine and can provide this benefit. Sorry for the quality of the images. So with this battery of behavioral assessment, we observed that those deficits that were present in males and females, okay, we tried to correct this, what we call endophenotypes of the GRIN2B loss of function model. And we observed that there was an increase of the general activity. These mice exhibit hypoactivity. Also, motor coordination was improved. So if you compare the GRIN2B treated with vehicle versus the GRIN2B treated with spermidine for 2 months, we observed this amelioration, and also in this rotor rod test. And very interestingly, we also performed experiments in the novel object recognition, in which we identified learning and memory deficits in this mouse model, and we observed that those, the mutant mice treated with spermidine did not show the significant differences with the wild-type group. So this was correlated with an improvement of the long-term potentiation evaluated ex vivo. And then we moved with all this data, we went back to the clinicians, and we showed all the data, and they started a proof of concept trial with 2 patients. They were 4 and 14 years old, respectively. And basically, they evaluated the potential presence of side effects or adverse effects, and also the potential benefit in terms of adaptive behavior. So in this spider plot, you can observe for case 1 or case 2, the dotted lines show the different dimensions that were evaluated in this adaptive behavior scale. And after the treatment with spermidine, we observed that for almost all the dimensions, except perhaps the written skills, there was an improvement in both patients. So this was published this year, and I will end up with the conclusions, okay? So I think that it's very important that GRIN variants need to be stratified, and we need to develop new methods for especially those complex variants. We have released this GRIN database that is being added to the community. We are currently developing this version 2.0 of the functional annotations. We have identified these 3 novel allosteric modulators, and hopefully in the next conference I will show you like proof of concept clinical trials, but let's see. So these are safe compounds also, not worthy. And we want to complete the evaluation in preclinical models and to translate to the clinical practice. And also, and importantly, we have developed this characterize the effect of spermidine for GRIN2B loss of function. And of course, we are following this study with a large cohort of patients, and this is something that Angels will also explain to you in the next conference. So thank you very much. Thank you very much, everybody, for the patience, and special thanks to people in the lab, Alba and Laia, young PhD students of Ana Santos, Mireia Oliveira, my right hand, and the clinicians. And of course, thank you very much, all of you, the families and researchers and clinicians. Thank you.